We have a series of lectures on methods in neuroendocrinology, and we start first today with some rather basic, we might say more antiquated methods, but I really don't like to refer to any type of method in such a way because we find that they are essential at some point, even when we use the most contemporary methodology. They are essential methods. Even though they might seem a bit obsolete and archaic, they are essential methods to get back to the level of the organ and the organism to show that there really is a function that meets the hypothesis that, or the set of hypotheses that a series of research scientists or a research group that has been working on, maybe just at the molecular or genetic level. So it's very important to keep these in mind. So we start first with some basic methods in just thinking about what we can do to assess hormones and their receptors and the physiological systems within which they're all working. So let's switch over to the computer, and we start with the methods in neuroendocrinology 1A. And we are considering the characteristics of assays from bioassays to cellular localization methods. And the bioassays are what I was saying are really the essential methods in endocrinology. Chapter 14 in the textbook covers discussion on behavioral bioassays that I think is excellent. You'll see that it is one of the chapters referenced for this series of lectures. And then there are a few other chapters that are referenced, and in those chapters there are just small segments that discuss this method or that method or a methodological approach. What we want to do is take some time to look at methods just in general, and this can be quite extensive list of methods, but we'll keep it somewhat limited. As well, we will add some when we get to discussions of the neuroendocrine axes in greater detail. I know you might think that we've been quite a level of detail so far, but it's still generic for all of the neuroendocrine axes. Of course, using one or the other as an example along the way, but we will take the detailed generalizations and concept collection that we have developed through the first dozen chapters and begin to apply that to each individual neuroendocrine axis as we proceed through the remainder of the semester. So the bioassays come back to the basic essentials, as I had said, and then we'll look at some cellular localization methods as we proceed through methods in neuroendocrinology 1B. The original file for this was 8 megabytes, and when I originally had put these together before any modifications, the computers in the lecture halls took about 20 minutes to load those PowerPoint presentations, so I split it into A through D and then methods 2, A and B, and that's the way they're posted on the WebCT. So we start first with asking some particular questions about assays collectively, and it's having to do with the factors in the validation of hormonal assays. So these are some basic terms that we would apply, or criteria. Criterion is singular, so each one is a criterion. It's a list of criteria, IA is plural, and a very brief definition. And these are very important because part of what we're proceeding to is the primary literature, and we are looking at the data in graphical and tabular format or photographic illustrations, and we start asking questions about these criteria to see how valid the data is, as well as whether or not it supports the conclusions of the authors in their attempts to contribute to our knowledge base in the area of neuroendocrinology, or neuroscience in general, in some cases that we use in this class. So accuracy is the first criterion here, And of course that deals with the nearness to true. So if you make a measurement, say of a hormone level in the blood, you know, if it is, if you get a value that is 100% factually true, that would be rare. So it's always within some range of tolerance. So any assay is going to have a tolerance in intra-assay variation. And we'll build on these ideas as we proceed. So you may run the same assay today and get one value on a sample, try to repeat the assay tomorrow on that same sample and get a slightly different value. So the method that you're using has some tolerance and your values will fall somewhere within that tolerance. If everything was perfect, it'd be purely accurate, but it isn't. So the closer to true, then the more accurate. Precision is the repeatability, so there's obviously an interrelationship between accuracy and repeatability, precision. So if you can't repeat it from one day to the next, you wonder whether you're truly getting an accurate measurement. But is it accurate enough and precise enough to be used to achieve the objectives of the study? Obviously sensitivity, so a minimum detectable quantity. Some assays may not be able to detect small enough quantities, let's say for example of hypothalamic releasing hormones in the plasma. There are levels of hypothalamic releasing hormones in the plasma, but they are extremely low and below the threshold of sensitivity for most of the assay methods. So a baseline by the method that wasn't very precise, wasn't very accurate because it can't even detect it, would be 0, undetectable levels. And then if you intervened with the system or perturbed the system in some way such that you got a dramatic dumping of the hypothalamic hormones into the plasma, then it would come into measurable range. You would still be able to say that even though you didn't have the sensitivity to detect the actual baseline, whatever you did obviously increased the amount in the plasma. But sensitivity is extremely important in many of the techniques that we'll be talking about. And then obviously specificity, the ability to exclusively discriminate a single entity. So how do you know what you're looking at is truly what you're looking at when you're making a measurement? So I use an antibody to luteinizing hormone to detect and quantitate luteinizing hormone in the plasma. How do you know the signal that you're getting from your enzyme assay system is only generated by luteinizing hormone? How do you know your antibody isn't binding to something else, maybe like albumin or something like that? So this is an example of specificity. We've seen in some of the previous lectures, like on chapter 9, that different molecules can bind to an individual receptor. So if you're running a receptor assay, you might run into similar situations. How do you know what you're detecting as being bound, or what you're detecting that has bound what you're added that you can measure, like a radioactive ligand, truly is what you are expecting to look at. So the measures to demonstrate the degree of specificity can be extensive. It will be a bit beyond this course to deal with that, but we'll show it in some cases. And then there's two other aspects of assays that we need to keep in mind. We talked about measuring LH levels in the plasma and the receptor binding quantity in our discussion of specificity. And those would be analytical assays. We're coming up with a quantitation of some entity. So we are taking an analytical approach of saying, what will it be at this time point, or after I've done this, or before and after I have made this manipulation. Maybe sequencing the DNA for a promoter region. That's an analytical assay. Gives you no functional information whatsoever. So some type of a bioassay or functional assay is essential to show that there is a biological significance to what is being measured in the analytical assay. In Chapter 9, one of the abstracts from a recent publication that we evaluated not only used an immunoassay to measure LH and FSH in the plasma of humans, but also performed a functional bioassay to show that their analytical assay, giving them certain quantitations, translated over to a similar change in the degree of a functional response within a biological system. So the reason for discussing this is that we'll see some research groups that collect data and simply quantitate certain things go on to draw conclusions that are very functionally based, and their data cannot support that type of a conclusion. So we're obviously trying to develop many skills and knowledge sets in this class. We want to be able to improve our ability to evaluate the literature critically, to evaluate textbook content critically. We want to be able to assess the value of methods and methodological approaches and their limitations, how that relates to drawing conclusions. And of course, we're trying to learn as much as we can about neuroendocrinology and obviously the methodology involved in these types of studies. And most of us know that much of this carries over to many other areas of investigation, possibly even if you're doing microbiology. You may not be measuring a hormone level with an antibody, but you might be measuring some other type of chemical signal within or between bacteria. Certainly, there are a lot of approaches of looking at enzyme assays and protein levels and so on and so forth. So looking at table 7.4 from the textbook, methods used to identify cellular sites of hormone synthesis, storage, and secretion. So we see that we run down this list very quickly. Identification of neuroendocrine cells at the gross anatomy level. So this is basically the naked eye, dissecting out the location, size, vascularization, and innervation of glands. Light microscopy, identification of the neural and the endocrine cells, not with the naked eye, obviously with a microscope, but there's many different microscopic methods that are available. Some of them now that are very dynamic, where you can monitor living cells, typically in culture. Really fantastic light microscopic visualization methods. Electron microscopy to identify the subcellular structure in the neuroendocrine cells. So typically these are freeze frame types of investigatory tools where you have to stop all life processes and process the sample such that it can be examined either with the naked eye, gross. Of course, you can perform surgeries and get survival surgeries and get gross anatomical information, but that doesn't provide very much investigatory information. But with light microscopy and electron microscopy, it's obvious that you are terminating the experiment, you're collecting a sample, it's freeze frame, so it's hard to place that type of data back into a completely dynamic process. At that point in time, under the conditions in which the sample was collected, this is what was observed. For purely descriptive purposes, it's very useful. For quantitative approaches, the basic light microscopy and electron microscopy still are somewhat useful, but they have their limitations. I recently visited a collaborator's laboratory in Denver, Colorado, and they showed me some renal cells that are targets for antidiuretic hormone, and they were tracking transport proteins in the apical membranes. These are long microvilli at the surface of these cells, and they actually had transfected those cells with what's called a fusion protein. It was a piece of DNA that had this fluorescing protein gene sequence attached to this transport protein, and thereby you could illuminate and activate and then pick up the fluorescent signal. So I actually saw in the living cell, when a stimulus was added, these membrane proteins were internalized and tucked away into vesicles deep within the cytoplasm of the cell. And then when you change the media again, so that it would reverse that, similar to the receptor downregulation and upregulation that we talked about. However, this was a transport protein. Then you would see the fluorescence come right back into the surface membrane of these cells. It was really fantastic to see it real time and live, and it was at the light microscopic level. So there are various types of methods at the light microscopic level today that do allow you to perform functional assays that are really quite elegant. The scanning electron micrograph, the confocal microscopy was part of what I was just discussing. They actually had to use two systems. They could see the membrane surface structures with one microscopic system, then they had to take the cells over to the confocal that could focus deep inside the cell and show them the vesicles. But when you put the two together, you get the same story. The localization of hormones within cells, so the histological staining can be general, but typically will include antibodies for immunocytochemistry, or can use fluorescence or enzyme labeling, histochemistry. There is a difference here in these terminologies. Immunocytochemistry, as the cyto implies, this would be the study of cells, the cytology of cells, typically for endogenous substances. And histochemistry, as histo prefix means tissue, it's studying the tissue in general. So there's subtle distinctions between those two terms, but not a big difference. And we'll talk about this method in a bit more detail, as well as a method for detecting messenger RNA within cells for verification of which cell populations actually express genes, and even semi-quantitative analyses. Examining the biosynthetic pathways, antibody injections. These can be used to detect and isolate specific molecules, but also when working with peptides and we don't have any type of antagonist to block their biological activity, you can inject an antibody or have it in the medium if you're working in the media in a tissue or cell culture system. And the antibody will bind the peptide and prevent it from interacting with the receptor. So it kind of acts as an antagonist in that way, but it's not a real one by the strict definition of the term antagonist. But I think you understand what I'm saying. You can still show that you can block the specific effect by inactivating the peptide. And that's a method using the antibodies. Obviously, drug injection, radioisotope injection, enzyme assays in general. We saw this in Chapter 9 in investigating the presence of aromatase in the anterior pituitary of fish and mammals. If you add testosterone, radio-labeled testosterone, can you isolate from that system radio-labeled estrogen and thereby verify that aromatase activity was present in the tissue sample? So radioisotope, radio-labeled molecules is very useful in endocrinology. So determining the chemical structure of hormones, chromatography, it'll separate the proteins into different fractions. Now there's gas chromatography, mass spectrometry, or GC mass spec, that is very useful for identifying peptides. For larger proteins, you can use electrophoresis, the method called Western blot that we'll describe as well. And amino acid sequencers, DNA sequencers. These are very important. We've already examined a lot of information about amino acid sequences of molecules, the peptides. We know which ones are octopeptides and tripeptides and things like that. But also we saw that the ligand-dependent hormone receptors, transcription factors, specifically ligand-dependent receptor transcription factors, are separated into two classes primarily by their amino acid sequences. So we all know that amino acid sequence and DNA sequence and RNA sequence methods are extremely important for gaining knowledge and substantiating facts in endocrinology and neuroendocrinology. So now let's begin to think about some common techniques in studying behavioral endocrinology. Throughout the semester we've talked about this coordinated psychogenic, behavioral, visceral through the autonomic nervous system, and endocrine responses to perturbations of the external or internal environment. So we're not spending a great deal of time this semester on behavioral neuroendocrinology. I did refer you to Chapter 14 where you can look over some of the information there. But we do recognize it. And we include it because we said from the beginning of the semester that we don't want to ignore the target cells and organs of these hormones and the contribution to the survival and reproduction of the organism, just to focus on the regulatory mechanisms of the neuroendocrine axes. So this is one opportunity for us to gain some appreciation of how some of these functional impacts on the organism can be investigated. So one of the major approaches still used, but used early on to determine the role of a chemical identified as being produced and released into the plasma where did I go wrong with that sentence? I forgot where I was going. Basic point I was trying to make was if you identified a chemical coming from an organ and that the chemical got into the blood and possibly was doing something, you take that organ out, chemical should be gone, and then you see what effect it has on the organism. Of course, what we see in especially the studies of the hypothalamic-pituitary gonadal axis, it's very common to remove the ovaries or the testes to produce a gonadal steroid-free animal, and then you begin to manipulate it so you've got control over those levels, and you can ask particular questions about the roles of those steroid hormones in certain functions within the organism, including behavior. So the removal of an organ is called extirpation. At this time, in the fall of 2005, we've had hurricanes and tremendous earthquakes around the world, and we hear the term extirpation to extirpate the victims that are trapped under the rubble, and it simply means to remove from the location wherein the individual or the entity resides. So extirpation of an organ is the removal of that organ. So removal of the gonads in the female, the ovaries, is an oophorectomy, O-O-P-H-O-R-ectomy. Removal of the gonads in the male is an orchiectomy, O-R-C-H-I-O-ectomy, E-C-T-O-M-Y. So an oophorectomy or an orchiectomy are gonadectomies in female and male rats, respectively. Removal of the uterus from the female is a hysterectomy. So we know that human females commonly undergo hysterectomies, and we, at least in general public, rarely hear the term oophorectomy, but a complete hysterectomy is both a hysterectomy and an oophorectomy to remove the ovaries too. That obviously is not for studying the effects of the hormones, but in a female who undergoes a complete hysterectomy, including the bilateral oophorectomy, will experience significant behavioral and physiological changes from the immediate removal of estrogen from the plasma. So they also would undergo a replacement therapy if it was medically appropriate. So extirpation and replacement is nothing new under the sun, but it is definitely a method that is continued in studying endocrine systems in the intact organism. Well, the whole organism. If you remove a gland, they're not intact anymore, right? So the extirpation is used to remove the organ as a source of the hormone. Then you ask questions about the hormone, either by replacing it or not replacing it, and then decide what you will observe. Bioassays, of course, are the test of the effects of the hormones on either the living animal, living tissue, or live cells. Immunoassays, we're moving now into the analytical assays. We'll talk about radioimmunoassay and enzyme immunoassays, even some receptor assays, immunocytochemistry, as we had mentioned, using antibodies to localize and maybe semi-quantitate molecules of interest, especially hormones and their receptors in the body. Autoradiography we've talked about already, using a radio-labeled molecule to create an image of where the radio-labeled molecule was located in the tissue after we terminate and collect the tissue and freeze frame it, if you will. Blot tests, we'll talk about Western blot, which is separating and blotting for proteins. We'll talk about the Northern blot, which is separating and blotting for RNA. We will not talk about the Southern blot, which is separating and blotting for DNA, but we will talk about Western blot and Northern blot. The whole cell or whole tissue homologue, if you will, or partner of Western blot is the immunocytochemistry, using antibodies to find the proteins in the cells. With the Western blot, you're also using antibodies to identify the protein and be able to quantitate it. And for the Northern blot, the homologue or the parallel system is in situ hybridization, a method that we'll discuss in the methods 1B. So we'll bring all of these different types of methods to our attention as we proceed through the semester to talk about the different types of data and methodological approaches to collect the data that generates our present state of knowledge about the neuroendocrine axes. As well, we can think about hormones on the level of the organism, and it doesn't just have to be behavior. It can be physiological, structural, as well as behavioral. It can have organizational or activational effects of the hormones. And it's very important to understand the distinction between these two. As we'll see, the activational is transitional, whereas the organizational is a permanent change that persists, although if you induce an organizational change developmentally in an organism that depends upon the presence of the hormone at the established state of that organizational change, remove the hormone and you probably will not observe the phenotype of the organizational change. So there's some subtleties to this, but still we can distinguish between them. The organizational effects of a hormone will be relatively permanent, as I said. They will be at least persistent, sometimes irreversible. And it's on structure and function of the body and depends on a critical period, a window of development of sensitivity. And of course, the most common critical periods we think about are adolescence. Shouldn't use the term adolescence here. Adolescence refers to psychosocial development through the pubescent time period. So if we're talking about the biological changes, we should be talking about pubescence here, not adolescence. So make that correction in your mind. And obviously, fetal development and embryonic development are critical time periods. And as I get older, I want to add senescence on the end of this as well. So pubescence and senescence and adulthood. There are still some organizational effects of hormones at that level. If you think of stress hormones destroying neurons in your hippocampus and impacting cognition, especially if you are more aged, then I see that as a window of susceptibility too. But these, obviously, the pubescence and fetal and embryonic are the time periods most commonly studied. The neonatal period or perinatal period, just before birth and just after birth, is also a critical window of time that is sometimes investigated. And we've seen examples already of investigations of developmental characteristics of the endocrine system. For example, the development of aromatase in one of the research articles whose abstract we evaluated. The activational effects of hormones, these are immediate. You inject it, it has an effect. The drug that you injected or the hormone is metabolized, eliminated, and things return back to baseline, homeostatic recovery. So these come and go with the presence or absence of the hormone. For example, synthetic steroid hormones is a prime example of that. So in continuing with our general assessment of methods, different types of assays, let's think about the spectrum of assay techniques. And we'll definitely get into more details as we proceed in the semester that will include focus on DNA. This one is focusing primarily on proteins. And we start first, of course, with bioassays that span from whole cells, living cells, dissociated target cells, to the slice of an organ, so at the tissue level, then the target organ, the organ level, then the whole animal, the organismic level, and then a group of animals, an actual experiment looking at maybe a population or a sample of a population of animals. If you're looking at intra-species behavioral effects, obviously you would have to have a group of animals. That's a prime example of the utility of this level of the spectrum of assay techniques, bioassays. If we go from bioassays in the other direction, of course we can begin to evaluate protein binding assays as well. So some of this might apply to whole cells, but typically to cell fragments or membrane receptors, as we saw with the study in a couple of graphs on the binding of corticosterone to brain tissue. It was the membrane fragment that was isolated and evaluated. Solution of receptors, so the receptors can be dissociated, proteins can be dissociated from the membrane, where we might get whole cell homogenate and look at all the receptors of the cell on the cell membrane, from the ER, from the vesicles, from the Golgi, all together. And of course can study solutions of antibodies. So as I said, there's the focus on protein binding assays in this illustration, but we can add in RNA and DNA binding assays on here, and we'll talk about some as we proceed in our discussions of methods and the value to gaining new knowledge and strengthening concepts and theoretical frameworks in endocrinology and neuroendocrinology. So let's consider some more characteristics of an assay. It should be specific for the hormone and physiological function of interest. So if you are studying thyroid stimulating hormone, you obviously are not going to use the gonad or the liver as the target tissue or organ to do your bioassay. It depends upon the question you're asking, but it seems obvious you'd be focusing on the thyroid gland to study bioassay of thyroid stimulating hormone. But where this becomes more important is we'll see that there are different cell lines, especially the anterior pituitary. We'll begin to see that there's all different kinds of permanent cell lines where we can study them in cell culture. One expresses this, one expresses that, might be the different tropes, somatotrope versus a lactotrope, and so on and so forth. It may also express a spectrum of genes, and their gene products are very important in the assay being run, the question being asked. And it may even be more so important that certain genes are not expressed, maybe in a signaling pathway, That way, if one of them is missing and you're asking a question about that portion of the signaling system, you can add that gene in to the cell line. So we'll see that this question is actually becomes more important when we start looking at different cell lines to ask different questions about the function of hormones and the relevance to hormonal systems. The assay should have good precision over a large working range, both within the assay and between assays. So basically what precision is related to is variation. So I'm just going to switch to the document camera here for a second really quickly and illustrate a bell-shaped curve of the frequency distribution of data. So we know that our data points, if we're doing good work, should be distributed in frequency in a bell shape with the mean, X bar here for our sample, and dispersion around that measurement of central tendency, or the variance, as we know. Standard deviation and variance are related. Variance is the square root of standard deviation. So with a good assay, these numbers are going to be, this curve is going to be tight to the mean. And this is a measurement of precision. So if that curve was horrible, rather than being tight like that, what if it was all stretched out like this, but had the same mean, and this is your variance? So you would say that this is not precise. And we can convert these two to bar graphs. If these were two bars, they may have the same mean, but our first curve that is more precise is going to have a very small error bar on it. And then the second one that's not precise is going to have an error bar. It looks like that one's going to span just about the whole height of the column. So obviously when we look at this, we picture data distributed like this, very widely dispersed around the mean. It is not precise, so this is not a good measurement. It is not precise. So that's basically what we're talking about when we are considering the precision. And so you want a low variance not only within each measurement that you take, each time you run the assay, you want to be sure that when you run it from day to day or morning to afternoon, that you have controlled variance or the same degree of variance excuse me, from assay to assay. So within an assay, this is called intra-assay variation, and there are methods to control that. And between the assays is the inter-assay variation. So it's very important to have standards and controls for intra-assay and inter-assay variations. And we'll see a few examples of this, but I think it's obvious that if you ran replicates in one assay and one gave you 10 and one gave you 1, your intra-assay variation, you ran them side by side at exactly the same time with the same reagents, your intra-assay variation is unacceptably high. If you did it morning and afternoon, morning you run it, you get 10, afternoon you run it, you get 1, your inter-assay variation from assay to assay is unacceptable. So that's kind of the example. So the assay should be capable of high throughput and should not require the use of highly specialized equipment. So the machinery, the microscopes that I was describing to you that allow you to visualize living molecules and living cells cost about $250,000. When you combine it with the confocal microscope to look at the intracellular components, you've got about $500,000 in equipment. So this is the point being made here, that you should ask most of your questions that don't require that degree of highly specialized equipment. It's the most refined and high impact questions that should be asked with that type of equipment. Otherwise, the most simplistic approaches make some of the most significant contributions because of the high throughput that you can get with it. You can put through, thousands of samples and collect, tens of thousands of data points to be analyzed in the same length of time it might take you to process one sample to get one data point with a very sophisticated piece of specialized equipment. The assay should be sensitive enough. I'm not saying don't use specialized equipment, but obviously you understand the idea of generating large amounts of data to have the high impact just on volume of data available. So the assay should be sensitive enough to detect the hormone of interest in normal and subnormal concentrations in the plasma, preferably without the need for extraction. That's referring to extraction of the hormones from the solution. So if you can only get a response by extracting the hormone of question or the molecule that you're suspecting to be a new hormone maybe from your sample source, and you have to concentrate it down and then add it to your assay so that the levels are way above the highest physiological level found in any biological system, then you're probably not studying something of any significance, or your method, your assay is completely inappropriate. Again, what's most important is we understand physiological mechanisms for endocrine systems based upon studies that use physiological concentrations of certain molecules. So the way that we can think about this is that there's subphysiological or subnormal levels or concentrations of the biologically active molecules. There's normal concentrations in the physiological range. There can be high supraphysiological levels. Then we get to pharmacological levels. And then finally we get to pathological or toxicological levels. So this is very important because when you look at some studies, they may be using a 100 millimolar concentration of the chemical under study. And when you look at some of the kinetics and the dynamics, should only be in the millimolar or nanomolar range to produce most biological effects. The data is highly questionable because why is it you can only get a response in the millimolar range when it should be in nanomolar or micromolar at most? There may be justifications for it, but we should ask those questions. And that's the importance here. So are you seeing with the high millimolar concentrations, is that just a pathological or a toxicological effect? Or is it pharmacological of a potency greater than even the highest physiological levels? Or is it well within physiological ranges of the drugs and the drug effects? Well, this slide is showing us some examples. It's panel A that's probably the most common one that you might see even in some of the papers that we have seen with the hypophysectomy, removal of the pituitary gland to study effects of hormones on the central nervous system. That's one of the figures from our textbook in Chapter 9. And giving that lecture, I asked the question, I don't know why they had to hypophysectomize to eliminate the effect of the target gland hormone, the gland 3 hormone, on the brain. In that case, I think it was the adrenal glands. But just the same, the hypophysectomized animal, it's possible to study the whole animal anatomically and the organs as well. And we see that the organs, glands 3, decrease in size because they don't have the pituitary hormones to sustain them. So this is still a rather common bioassay approach in the whole animal. If you were to use these glands and their cells, tissue slices or cells to perform studies, you would be studying very different tissues and cells, because these would have been starved from the pituitary hormones. Comb size in chickens, the roosters can be studied, and antler growth in deer. So not all of us might end up just studying rats or mice in the laboratory. Some may actually go into the field for environmental studies and maybe even help us find a chicken that grows fast and doesn't get the avian flu virus, right? So the study of the hormone effect production, we said the extirpation, the surgical removal of the gland from the body. Castration is an example. So we'll see that repeatedly in the literature. Remove the testes from the male species. This will show the effects of not having testosterone in the body, obviously. It's not fatal, can cause both activational and organizational effects on the developing male and the adult male. So castration will have little effect if done after puberty, as we know. It has mostly activational types of effects, changes in behavior, but it doesn't eliminate the behaviors that are supported or strengthened by the presence of the gonadal steroid hormones. So the hormones like testosterone will have different effects if removed or added at different times of development. Castration in humans will have the following results if performed at a critical time during development. So early in life, short stature, long limbs, no secondary sex characteristics, no beard growth or vocal maturation, no libido. Whereas after puberty, there'd be very little consequences, maybe a loss in sex drive, and typical male activities like football and power tools. Big smiley face there. I think that's just a joke. So we continue with these questions about extirpation because I want to take you to a few images where you have to guess, make an educated guess and predict where the interruption might have occurred. So in certain animal studies in the early 1800s, Arnold Berthold studied the chickens and castrated the baby male chicks, producing the capon that had no comb, no wattle. It was smaller, more tender meat, no crowing, no fighting, no mating behavior. So obviously this is a better way to generate commercial chickens. So 3 treatment groups with the results are shown here. You remove both testes and it becomes the capon. Reimplant the testes in the stomach cavity at different location. The rooster develops. The rooster with 1 remaining testes that is 2 times as large. You remove both testes and reimplant only 1, and this is called compensation. This can happen. Removal of one of the organs, and the other organ will enlarge and compensate and be responsive, and it should not have any major detrimental effects. So in that last control group, the testes grew twice as large due to a process called compensatory hypertrophy, an example that's detailed in this series, the last couple slides that we'll take a look at. So in this series of slides, we see the graph on the right. It's just a hypothetical graph of the plasma levels of the hormones. In this case, I'm going to get kind of close to look at it. It should be GnRH, LH, and testosterone from the male. So the behavioral effect, there's no hormonal normal activity and hormonal production in this. And of course we have the GnRH from the hypothalamus to the median eminence for the release at the neurohemal junction, the anterior pituitary gonadotropes for LH synthesis and secretion, the LH targeting the testes to produce testosterone with the negative feedback. So this is our functional circuit. We switch over to this slide and we see the question, what has happened here? And we determine that the only of these 3 hormones that we see for the HPG axis is testosterone. So we ask the question, what has happened here? How could we lose the LH from the pituitary and the GnRH And of course, the answer to this question is that there is excess or extra testosterone exogenously added as an anabolic steroid. So in individuals that take anabolic steroids, either clinically or illicitly, it will act at the anterior pituitary and the hypothalamus to cause negative feedback and silence those if the high enough doses are given. And then the only testosterone seen in the plasma excuse me, would be the exogenous testosterone, because the gonadal source would be lost. And this is, of course, why in individuals that discontinue the use of the anabolic steroids, their gonadal function, their HPG function, remains suppressed. And then here we have another one. We see we've got GnRH, but no LH and no testosterone. We ask what has happened here. And of course, this is extirpation of the anterior pituitary, the hypophysectomy. I'll just skim through these last set of slides at another time. I'll include them in another lecture. It's just ways of looking at graphical data that I'd hoped to have time to introduce at this point. So that completes our discussion of methods 1A. I appreciate your time and attention and hope that you have a nice day.
Thank you.
Bye-bye.